Welcome to the Biological Life Sciences channel, students. Today we will be seeing how we can analyze a PDB file. Okay, so let's see one by one. Okay, first of all, for analyzing a PDB file, you have to open the protein data bank. Okay, so protein data bank stores all the 3D structures that are available, which are determined using X-ray crystallography or NMR studies. Okay, so now let's see how we can download a sequence. So this is the home page of PDB. You have to put a URL as www.rcasp.org, and when you press enter you will be going to the website of homepage of protein data bank. Okay, so this is how it opens. Now in the search bar you have to put any example here. For example, you can search it by PDB ID, you can search it by ligand ID, sequence, or you can put a keyword also. Okay, so I am putting a keyword as myoglobin. Okay, okay, so I am putting a keyword as myoglobin. Okay, so when I put myoglobin and search it, it is going to show me number of results. Okay, you can see the number of results that is being shown here. So these are all related to the myoglobin. Okay, so for example, if I want to analyze the first file that is having a PDB ID 5 MBA. Okay, so what I am going to do, I am going to click on that file. Okay, so that file will be opening in front of me. So you can see here this file opens. The classification of this protein that I searches is oxygen storage. So as we know, hemoglobin is responsible for transport of oxygen or storage of oxygen. So this is how the 3D structure looks like of the myoglobin from organism called as Asplicia limkina. Okay, so let's now download the file. So on this menu you can see here download files. You have to download in legacy PDB format. Okay, so when you do this, the download will start and file will get download. Okay, now this file gets opened into your download folder. You have to cut it from here and just paste that file onto your desktop. Okay, so when you paste that file onto your desktop, see this, you can here you can get this file. This is your file, PDB file. Okay, now for visualizing that particular PDB file, you have to download a software called as RasMol. So we are going to see how we can do the structure visualization using RasMol. Okay, so let's see or download the RasMol first. So you have to put the RasMol name into the Google search. The RasMol software's link opens up first. Click on that link, you will go to the home page of RasMol software. You have to see here different setups of RasMol software is there. RasMol Windows installer software, you have to click on it and you are going to get a setup file for RasMol. Okay, sometimes due to some errors the file may not download. Just proceed in a unsafe mode. Now this setup file downloads here. Double click on that setup file. The computer will ask you to install that particular software. So proceed with English as a language, agree to the conditions given and install the software. Okay, so you can see now here we have installed the RasMol software. Okay, so press close button, and the icon that RasMol software looks like is this. Okay, it is called as RasWin. Okay, so you can, how to open this PDB file in the RasWin, this 5 MBA file, just drag the file onto the icon of RasWin. Okay, so this opens up. Okay, you can see here the RasMol has two interface and our file has opened into that database. Okay, so I'll slightly enlarge the file so that you can see it properly. Okay, so this is the RasMol window, you can see here, and the other interface of RasMol is the command line interface. Okay, so you can see the smaller window. This is the command line interface for RasMol. Okay, so whatever command you put here in this command line interface, you will get all the result in the displays interface. Okay, so there are different commands that you can opt or you can open for the RasMol purpose. I'll show you some commands so that you will be able to open that, or you will be able to visualize the protein in different modes. Okay, so let's see some of the commands. So first of all, you are getting certain information here. You can see you are getting certain information about the protein structure. Okay, but if you are not getting the information, what you have to put, just put show information. Okay, so when you press enter, you are going to get every details of that. In case you are not getting the details when you are opening the file, PDB file in the RasMol. Okay, so show information, if you put, you will be getting the result. Okay, now remember here, the command that you are typing is very much important here. You have to strictly adhere to the sequence or the spaces or the capital letters and all that you're typing here, because it is case sensitive. Okay, so now I have executed the first command here, that is the show information command. So I have got all the information about the myoglobin molecule that I have opened here. Okay, so this is the structure of the myoglobin molecule that I am able to see. Oxygen storage record, PDB records, then the ID of that particular molecule, then the technique through which the structure was reduced, the number of chains, groups, atoms, bonds, helix, etc., etc. Okay, so if I now, let's execute the next command. So if I have to see the sequence, I'll put a command as show sequence. So I'll get the sequence. Okay, so there is chain A, which is having so many number of amino acids, about 146 amino acids. You can see in the this summary also, 146 amino acids are there. The last one is the alanine amino acid. Okay, so this is how you are going to get a sequence. Okay, if you want to execute the next command, then you can put the command as select all. Okay, when you press enter, all 1258 means 1258 atoms in this structure will be selected. Okay, so this is how the commands are executed. Now let's see certain display commands. So if I put here certain display commands, okay, now there are lots of display commands available here. Students, these display commands are available in the menu here also. All these commands are there. You can see wireframe, backbone, then sticks models, space fill, ball and stick model, ribbon, strands model, cartoon, molecular surface. So when you click on that, you can see the structure changes. Okay, so even if you want to change the structure using command line, you can do that. Okay, so this commands or this structure is also, you can rotate this structure also. Okay, so this is how it is done. Okay, now let's see how we can do change the structure using command line. So I will put ribbon. So when I put ribbon and enter, the structure opens in the ribbon form. Okay, then I'll put wire frame. Okay, so the structure opens in the wire frame. Okay, then again backbone, suppose, let's see backbone. So the structure opens up in the backbone. Okay, so this is how you can use different commands. Okay, so let's see strands. So you can see the structure in strands command. Okay, so this is how you can execute various display commands. Okay, now let's put cartoon. So this opens up in the cartoon command. Okay, so this is how you can execute various uh, functions in the RasMol. Okay, now let's use certain restrict commands here. So restrict command will restrict the structure that we want to see. Okay, so let's do it. Okay, so first of all, let's put restrict helices. Okay, so only helix in that particular structure will be restricted. You can see all the helix that are only shown to you here, other structures are not seen. Okay, and helix contains 856 atoms here, that is also shown to you. Okay, now let's restrict sheet. So there is no sheet in this structure. See, so there is no atom selected, so no sheet. Okay, then if I want to check whether H bonds are present, H bond, I'll type H bond and press enter. You'll get 118 H bonds. Okay, okay, H bonds. Okay, so now let's put the restrict command again, so that we can see the structure. Restrict helices. We are not able to see the structure here. So okay, okay, so this is how the structure looks like. Now H bonds, how many H bonds are seen here? How many H bonds are seen here? You can see there are 114 H bonds. Okay, so hydrogen bond you can calculate. Then you can calculate the SS bond also. Okay, SS bond means disulfide bridges, that plays a very important role in See, you have to be very careful with the typing, because it is case sensitive. Okay, SS bonds. So there is no disulfide pressures here in this structure. You can see it is shown to you as 0. Okay. So remember, in H bonds and in SS bonds, you don't have to put the gap here. Okay. So don't put this space. Okay. So you have to just carefully add the commands very carefully in the command line window, and you will get the results as operated, or the as you can see the result here in the main window. Okay, so this is how you can carry out different structure visualization and different analysis of protein structure using RasMol. Okay, then you can restrict certain atom. Okay, so let's see, by putting that restrict glycine. Okay, so some error it is showing, select glycine will type. Okay, oh, single letter code you have to put. Single A, three-letter code. Okay, so see, only glycine is selected. I have put the command as select glycine, GLY. That is the three-letter code for glycine. Okay, so it has selected only the glycine in the structure, and it is showing me that glycine atom, glycine amino acid is having 44 atoms in this structure. Okay, so this is how you can visualize a protein structure in RasMol. I hope you will be able to visualize or do the experimental analysis of such kind of structure using RasMol. Okay, so thank you for joining today's video, students. Thank you for listening.